Okay, welcome everybody to the seminar. It's a great pleasure to introduce Clare Mackay. I think most of us know Clare very well as the professor of imaging neuroscience. She works at the Department of Psychiatry and is one of the leading figures in the BRC, especially the new application. Today, she will focus on her new project that I at least thought was very interesting, at the Oxford Brain Health Clinic, and will present the first findings and a description of the pilot data for us. Clare, thank you for presenting, and the floor is yours.
Thank you very much, Cornelia, and to Nick and Masud for inviting me to talk to you today. I'm very happy to be here to tell you a little bit about our new brain health clinic, and I've got some very early results from that that I'm looking forward to sharing with you. I thought I would start though just by giving a little bit of background to the sort of work of my group over the years, so that you can see sort of the context of where the brain health clinic came from in my journey, and then why I think it's important in the larger context. So my team and I are very much team scientists, and so I've been very fortunate over the last decade and a bit to work as part of some of these very large sort of infrastructure and scientific programs that have been led by my colleagues around Oxford. And I'm going to give really brief details about some of these, just by way of introduction, really, before I spend the majority of the talk on the Oxford Brain Health Clinic. So this is a cartoon of a person's lifespan, and you can choose whatever your favorite function is. It could be motor or cognitive up the y-axis, and then we've got time along the bottom. And we'd like to think that we reach a level of performance in our early adulthood, and that is maintained through life. Of course, for most people, depending on the function, there's a degree of reduction in our ability to perform that function as we get older, and an increase in the variability of people's abilities as they get older. For some individuals, this might veer towards a sort of preclinical impairment stage, and then if a particular threshold is met, you would describe somebody as being in the disease stage. And really the work of my group over the years has been is kind of around this cartoon. So first of all, what is normal aging? How can we characterize normal aging as far as brain health is concerned? How can we understand the risk for cognitive or motor decline? Are there mechanisms of resilience that can potentially be harnessed to preserve cognitive and brain health as we get older? Can we develop high quality biomarkers for experimental medicine? And then finally, can we use these biomarkers to improve diagnosis or prognosis at the clinical interface? So I've just got 3 slides, which are going to be quite a sort of rapid run through some of the work that we've done over the last sort of 15 years or so. So starting with ApoE4, for those who don't know, Apolipoprotein E has one of the, the best established risk factor for Alzheimer's disease in the form of epsilon 4. If you carry one version of the epsilon 4 allele or more, you are at increased risk of Alzheimer's disease. And ApoE is a really nice kind of model gene in that it also has a protective version. So if you're lucky enough to carry an E2 allele of ApoE, then you have a lower than average risk of Alzheimer's disease. And you can see on here the proportions of this gene in the general population, and so you don't have to screen too many people before you can find yourself a risk group and a resilience group. And we've done quite a lot of work on this over the last sort of starting from 15 years ago, when we discovered that using new fMRI techniques, functional magnetic resonance imaging techniques, we could identify differences in the functioning of brains in young adults who did or did not carry the ApoE4 allele. And so in particular, we found differences in the way the brain is behaving in the very structure, in the hippocampus. So in the very structure that decades later might be implicated in cognitive decline, we can already see differences in the way the brain is behaving with people in their 20s and 30s. So that really led to a body of work where we used various different imaging techniques in people who were characterized according to ApoE genotype over the age span, looking at both risk and resilience. And this body of work is really now the subject of Sana Sori's Alzheimer's Research, Alzheimer's Society Fellowship, and she discovered that cerebrovascular reactivity, so the way that the blood vessels behave in the brain when they're challenged, might well be the mechanism that gives rise to a lot of this fMRI differences that we see, and that's really the work that she's now leading on. And this is a picture of the ApoE gang from over the years. Switching now to Parkinson's disease, and again, I was fortunate to be to lead the imaging aspect of the Oxford Parkinson's Disease Centre, led by Richard Wade-Martins, and the clinical lead being Michelle Hu. Over 10 years, we tried out a number of different imaging techniques to try to identify potential biomarkers, and also to identify risk markers for early Parkinson's disease. Again, there's a big gang of people that were involved in this work over the years, and this is now really being led by Johannes Klein going forwards. And then a third slide of another big collaboration, which is the Whitehall 2 imaging sub-study led by Klaus Ebmeyer. And this was an opportunity some years ago now to scan, to do a high quality MRI scanning on a subset of the large Whitehall 2 epidemiological cohort. So this is individuals who've already had 30 years worth of information collected about their general health and lifestyle through their adulthood, and then are being imaged in sort of towards retirement age. And that's allowed us to look at all sorts of factors, diet, exercise, mood, alcohol intake, and cerebrovascular and cardiovascular risk factors, and look at how they influence brain health in later life. And again, lots of the members of Klaus's team have been successful in obtaining independent fellowships to sort of take this work forward in various ways. So hopefully you can see that there's lots of work that's been done there in relation to the risk and resilience and understanding normal aging. But personally, over the years, I've become sort of more and more interested in pushing towards the more translational end of this work, and really wanting to see how we can make the work that we're doing benefit the patients and the NHS. And the context of this really is on this slide. So within dementia clinical practice, as an imager, if you're wanting to interface with the clinical service, then you have to look at what the clinical service is doing and see where you might be able to have an impact. So this spectrum goes from, so there's a large number of patients who are diagnosed with dementia who never get any imaging. Probably the biggest fraction have CT imaging, which is then comes with a standard radiological report. If you're lucky, you might get an MRI scan, and again, the standard way of reporting that is a sort of text-based report. If you're very lucky, you might get a sort of standardized report with some semi-quantitative assessments being made. So a Shelton score is or an MTA score is a measure of atrophy. A Fazekas score is a measure of white matter changes in the brain. A very specialist clinic might be able to offer actual quantitative information like hippocampal volumes, but this is really already verging into a research environment where, of course, we can do all sorts of fancy schmancy things, but these are really not available to the masses in terms of the being able to translate things into clinical practice. So almost everybody is at the bottom end of this scale, getting either no imaging or CT imaging, and a lucky few fall into the top end of the scale. And so really for me, it was about, you know, as a research clinic, we're all the way at the top end, or research environment, we're all at the way at the top end. How can we bring some of this to a larger group of people? And that's how the idea for the Brain Health Clinic was born. So the idea is really very simple. We're wanting to create infrastructure to enable an integrated clinical service and research opportunities. We want to be able to offer high quality assessments as standard for patients who are coming through the clinical routine, and research assessments as extras with as little barrier as possible to the to access to those research assessments. And this, for me, really creates this translational interface, so that as we're developing novel ways of imaging, novel ways of assessing cognition, novel biomarkers, blood-based biomarkers, whatever it might be, then we have a way of quickly and relatively easily testing those in a real world clinical population. So I've described why I wanted to create a brain health clinic, but why does the world need brain health clinics? Well, memory clinic services are sort of widely understood to be outdated and not really set up at all to deliver disease-modifying therapies. Of course, we don't have any disease-modifying therapies at the moment, but it feels like they're not so far away now, and our clinical services are going to have to be radically updated in order to deliver these. There's also growing evidence that lifestyle interventions can be disease-modifying now. So the plot on the right, which is a little bit difficult to decode, but this is work from Jill Livingston in the Lancet Commission, putting together tons of studies, tons of evidence, a massive systematic review of the evidence, demonstrating that lifestyle factors or modifiable risk factors, if you were able to modify all of those risk factors, you could prevent or delay up to 40% of dementia. That's a massive potential impact. So, and that sort of potential impact isn't really being delivered to patients at the moment. So those are the sort of reasons from the clinical side. But on the research side, the majority of dementia research is particularly the sort of research that I've been involved in. So, you know, relatively small N imaging studies, or the sort of early phase translational research, neuroscience type research. It's often carried out in rather selected cohorts that don't actually very well reflect the population of patients that that we hope that it will one day be translated into. And of course, clinical trials are quite severely hampered by a lack of infrastructure. So we put together the ambition to develop a brain health clinic in our NIHR Biomedical Research Centre 5 years ago, and 5.5 years ago, when the Oxford Health Biomedical Research Centre was launched. This was one of the key ambitions, and the main ambition in the theme that I lead. We proposed to pilot the brain health clinic in 150 patients from the Oxfordshire South team, so that's South Oxfordshire, primarily, as well as the BRC, the Biomedical Research Centre. We have had both funding and huge amounts of support from the NIHR Oxford Cognitive Health Clinical Research Facility, also based up at the Warnford. And the pilot was really to test the feasibility. Can we set this up? Can we make it work? Can we get all of the nuts and bolts sorted out, the IT, the ethics, the governance, etc., in place? And then what does the proportion and profile of patients look like who can attend the brain health clinic? What are the proportion and profile of patients who consent to research? What's the experience like for the patients and the carers? What's the experience like for all of the clinical teams working in and around the clinics? And then what does this cost? Is it scalable? Is it sustainable, etc.? And I need to pay huge credit to Lola Martos, who's the lead for older adults in Oxford Health NHS Foundation Trust, who's been very much my sort of partner in getting this off the ground, and Vanessa Raymond, who is now the Director for Research and Development in Oxford Health. Again, as the three of us have really worked hard on getting this to work together. So this is what the current pathway looks like if you are an older adult above 65 years old and you visit your GP concerned about your memory. This is a sort of simplified version of the pathway that you will go through. So if the GP refers you into the memory clinic service, the psychiatry-based memory clinic service, there will be a decision made without seeing the patient about whether this patient should be referred for a scan. If a scan is deemed likely to be helpful, then they'll be sent for a CT scan en route to their memory clinic appointment. So the patient would go and have a CT scan and then come to the memory clinic and see the doctor after the radiologist report has been written. So we saw an opportunity to interact with this pathway. And so what we've done is ask for those referrals to come to the brain health clinic instead of going to CT. So we're able to offer MRI scans and as well as a cognitive and clinical assessment. And then by being able to link up with the radiology services, the data that we're collecting, the MRI scans that we're collecting go straight back into the clinical route as it was before, and the patient attends memory clinic just like they did before. So essentially we've bypassed the CT scan and added in a whole bunch of good assessments. We do this at OBER, the Oxford Centre for Human Brain Activity, which is part of the Wellcome Centre for Integrative Neuroimaging. And on Tuesdays, we turn this into the Brain Health Clinic, really, again, with massive help from the clinical research facility who are essentially both staff and help manage the centre so that it is in effect an NHS environment while we have NHS patients coming through. At this point, I just want to make a huge shout out to our PPI advisory group, who helped us put together everything to do with the Brain Health Clinic, including doing all of the walkthroughs and the early figuring out what the signs should look like, what the ethics documentation should look like, etc. We had a huge amount of input from our PPI group in putting this together. So one of the key aspects of the Brain Health Clinic is the consenting process. So everybody who comes through the door is asked if they are interested in taking part in research. They can say no, that's fine. We'll still give them the good quality assessments. And then they can say yes in any one of three ways. And we like it if they say yes in all of these ways. They can say, yes, you can use my data. Yes, I'm happy to do more while I'm here. And yes, I'm happy to be contacted about future studies and trials. And then depending on which they say yes to, the protocol then unfolds for their visit. And very broadly, the protocol involves the clinical and cognitive assessments, the research consenting process, and then the MRI scan. And then if they've consented to doing additional assessments while they're there, they'll do more scans while they're in the scanner. They'll provide a saliva sample, complete some more questionnaires, and do some research cognitive assessments as well. And the information is all collated so that on the NHS side, that we produce a report that goes into the electronic patient record. And then on the research side, we the data, where a consent has been gained, goes into the research database. The research database is comprised of the cognition, the genotypes, the whole bunch of questionnaires, medical history, diagnosis, etc., the imaging derived phenotypes. And ultimately, the destination for this will be the Dementia Platform UK's data portal, where it'll be freely available to people to interrogate. So how has it been going? So we spent, it took us a long time to get to the point of launching. We managed to launch in August 2020. For those of you who will, I'm sure, remember that was kind of in the middle of the first year of the pandemic. And so things got delayed, but nevertheless, we were able to open in August 2020. And most of the data I'm going to show you today comes from for the patients that came through the Brain Health Clinic between August 2020 and November 21. And at the point of which these slides were made, then 150 people had been referred. And you'll be seeing data from about 110-ish people who consented for their data to be used in the way that you're seeing. So the profiles of the patients coming through are kind of as you would expect. So mean age of these patients is in the late 70s. You can see the spectrum of cognitive scores and age and depression scores in the histograms below. But these are pretty, I think, for people who are familiar with the population coming through psychiatry-based memory clinics, this will look very familiar to you. We have got some really excellent patient feedback along the way. I won't go through this slide in detail. It's always wonderful to see this feedback, but we do of course try very hard to make sure patients have a nice time as they're coming through the centre. So it's perhaps not surprising that they feel well looked after. I'm very pleased that they feel well looked after. But a couple of things just to draw attention to is the blue pie charts in the bottom, which demonstrate that people indicate that more than 80% of people are indicating that they would have been happy to do more while they were in the clinic that day. And again, more than 80% are willing to do more relating to the clinic once they've gone home. So we're not wearing people out and putting people off doing research while they're with us. I've already shown a little snapshot, a little thumbnail of the clinical report that goes into the electronic patient record. But the real headline here is that due to the nature of the report that we're putting together, it's quite comprehensive. That means that when the patients get to clinic, their appointment time has been reduced from 70 to 45 minutes. So that's time in front of a consultant. So as far as the NHS is concerned, that's expensive time. So there is a cost saving to add into the equation for the NHS of having this sort of assessment outside of the routine appointments. One of the things that we were able to do along the way was to support a Declineside project. This is a qualitative research study of the attitudes of the clinical staff working in and around the clinic. And this importantly includes people who are sort of involved in the development of the brain health clinic, but also a whole bunch of the staff who were peripheral to that. So they're not invested in the brain health clinic already. And really the headline here is that the sort of pervading attitudes before the pilot took place involved comments like, a lot of our patients don't want to take part in research, and I don't think it's right to put patients through more than we already do. And then by the time the mid-pilot came along, the same people are pleasantly surprised that so many patients are able to tolerate the MRI scans and longer assessments. And we had lots of comments about how useful the report is that we're producing. So that's good. And then one of the real major headlines is the research consent rate. So this is against a national target of wanting 10% of patients with dementia to be given an opportunity to take part in research. So as you saw, we ask everybody who comes through the door if they're interested and willing to take part in research. 93% of them are happy for the use of the data that we're collecting to be used for research. 86% are happy to do more assessments while they're there, and 73% are happy to be recontacted about future studies and trials. Now, this is quite a big deal because, of course, we took the risk of offering this service without knowing what proportion of patients would consent to research. So this is really gratifying from my point of view. Digging down a little bit further, when we look at those additional assessments, you can see that the percentages of people who consent and then are able to complete the various assessments varies. So we have 63% consenting to research MRI, and then 43% being able to complete it. That doesn't mean that the other 20-ish% did no additional tasks. It's just that we ask them after every scan, do you want to carry on? So we give them lots of opportunities to say no if they want to. And you can see equivalent things there for the saliva samples and for informant questionnaires. And then speaking of the informants, this is the relatives who are accompanying the patients. A large proportion of them are completing additional questionnaires relating to their relative. And again, a large percent are happy to be recontacted about future studies and trials. So we think this gives us a really nice representative cohort. So since such a large proportion of the patients coming through, and again, these are not selected in any way, these are just people coming for their routine NHS appointments. A large proportion of them are consenting to research. So that means we end up with a nice representative cohort. What do I mean by that? So if we just look at age, for example, this is a plot of the average age of diagnosis for people with dementia. This comes from the large population registers in Sweden and Finland. So you're looking at data from, I think, more than 25,000 people here. And you can see that the average age that people get diagnosed with dementia is around 80. Now, for those who are not aware, in the UK in particular, we have a sort of specialty divide, which roughly is, or rather, age is one of the factors that largely influences this divide. So if you are concerned about your memory under the age of 65, and or you have an unusual presentation in terms of cognitive deficits, then you'll likely be referred into neurological-based memory clinics or cognitive disorder clinics. Whereas if you're over 65 and you have a more typical looking Alzheimer's-like memory impairment, then you'll be referred to psychiatry-based memory clinics. And if your memory problem comes to light as a result of going into hospital for some other reason, then you'll most likely find yourself in gerontology-based memory clinics. And as you can see here, I'm highlighting the fact that whereas in neurology there's quite a strong kind of research culture, there's access to decent quality imaging and neuropsychology, and follow-up is more standard. But in psychiatry context, the culture is less sort of research orientated in general, not of course not everywhere, but in general, there's less research going on. There's much less access to MRI scans. And so in my psychiatrist older adult colleagues tell me that they find it very difficult to get an MRI scan, even when there's good reason to ask for one. And they don't tend to have access to separate neuropsychological services. And at this point, the service is really set up around diagnosis, and so follow-up is less common. So you can see there's a bit of a problem here, because the great majority of patients are not getting access to the higher quality assessments or the follow-ups, etc., or being given as much opportunity to take part in research. And this sort of comes to light really when you look at the large cohorts on which most of the research is, or rather a large chunk of the research is based. So here I'm highlighting the average ages that are in the Amsterdam dementia cohort, a large cohort of individuals based on a very similar model. So the Amsterdam Dementia Centre collects together a cohort of all of the individuals that they're seeing clinically. So the Brain Health Clinic is kind of based on that same model. And then for the neuroimagers in the group will have heard of ADNI, the very large neuroimaging, Alzheimer's disease neuroimaging initiative. And I would say, I haven't checked this, but probably sort of 60 or 70% of all neuroimaging in dementia literature is based on the ADNI cohort that's been, and there are several ADNI cohorts over the years. But as you can see, the mean ages are a little way off the mean age of the total population. And so again, as an imager trying to develop neuroimaging biomarkers, we're not actually necessarily having easy access to the patients that we're trying to develop these biomarkers for. So I've roughly overlain on here what the Brain Health Clinic population looks like. And as you can see, it sort of nicely fills that gap for, so we're basing our clinic is at the moment entirely psychiatry based. So we're not seeing patients under 65 years of age, but the rest of the histogram looks very much like the background there. So what do the patients look like who are coming through our doors? So this is pretty hot off the press that I'm able to show you these plots. We've only very recently had access to the diagnosis, which of course come after we've seen the patients. So the patients come through the Brain Health Clinic and then they have their memory clinic appointment. It's only after that we have access to the diagnoses. So this is looking at 113 patients. As you can see, there's about a third have MCI, mild cognitive impairment, about a third have typical late onset Alzheimer's disease. But actually, if you need to add all of the kind of blue colors together to see the overall proportion of people who have established dementia. And then there are some individuals who don't have any diagnosis after their clinical appointment. That's the orange wedge. And then there's a few who are diagnosed as having mood or anxiety disorders that are likely to be causing their cognitive problems. So that's what the profile of patients is looking like at this point. And then we can also have a look at their ApoE genotypes, and this is pretty much exactly what you would expect from a memory clinic population. So the proportion of people with the E3 genotype in a general population, a non-demented population, that would be about 65%, and it's 50% in this population. And the proportion of people carrying an E4 allele is higher in this population than it would be in a non-demented population, exactly as you would expect. So one of the key things is about the extent to which these patients can tolerate MRI. Now, one of the kind of real USPs of our setup here in Oxford is that we matched our acquisition of MRI scans to the UK Biobank. I'm going to say a lot more about the UK Biobank in the next few slides. But our scanning protocol is divided into the clinical sequences here in blue, and then the research sequences in green. And the flowchart on the right tells you how the proportions of patients who are able to tolerate the scan. So of 108 patients who have been through the centre, 100, so almost all of them, are able to complete the clinical MRI scans. There are 95 who consented to the use of their data for research. So that's 88% in overall, and then 64% consented to additional research scans, and 44% completed all of those additional research scans, and then there'll be numbers in between who are able to complete some of them. So the 44% means, you know, we're less than half who can get through the whole protocol. But really importantly, even the 88% are having UK Biobank sequences. So the T1 and T2 images that we're acquiring are matched exactly with the UK Biobank. And so I've mentioned this a couple of times already, but I'm sure most of you know that the UK Biobank are more than halfway through now, collecting 100,000 high quality MRI data sets from the population sample that they have. And they're also repeating MRI now in 10,000 people who are coming through. The UK Biobank protocol was put together by a consortium led by Oxford, and includes all of the sort of the high quality imaging protocols that we've been used to dealing with over the last few years, and a few sort of specialist extras as well. But one of the things that I think, in a way, so collecting that many brain scans is already game changing. But the other equally game changing thing, which is again being led by my colleagues in Wynn here in Oxford, is what we're able to do with them now. So Steve Smith and colleagues have put together now an automated pipeline. So with this many brain scans, we can't do what we've always done, which is to be quite sort of hands-on and have to do quite a lot of things by meticulous, careful checking on an individual basis. That's what we imagers have typically done over the years when we've been dealing with numbers of subjects in the tens or maybe hundreds. But now that we're dealing with numbers of subjects in the tens of thousands, we need to come up with new ways of automatically extracting information from these images. And that's been the work of the Wynn team over the last few years, and it's now become kind of routine that these pipelines spit out more than 3,000 imaging-derived phenotypes that then go back into the biobank and are the UK Biobank and are available to be entered into analyses just like any other piece of data. So you have the first amazing analyses coming out where all of the imaging derived phenotypes are being looked at against all of the genetic information and all of the lifestyle information and all of the health information. So an incredible resource of an incredible big data resource, which is now we're just sort of beginning to know how to exploit for the benefits of translational research. So as I say, alignment with the UK Biobank means that we can immediately start to think about how that huge data set is useful in the context of individual patients in a very sort of real world clinical setting. So I've already talked about the work that we've done to align the protocols, but we also need to adapt those pipelines so that they work in this sort of clinical population. So the pipelines have been designed to work on a general population level, but we're obviously dealing with individuals who have quite a high degree of atrophy. They have a high degree of white matter hyperintensities. Their brains are a little bit more, as a group, they're more unusual than the whole group. And so my colleague Ludovica Grifanti is really now leading the work that we need to do to adapt the pipeline so that they are robust and reliable in deriving those same phenotypes in this clinical sample, and that work's well underway. And then, of course, we have the potential to use the population in order to be able to make inferences about our individuals. So here you're looking at something called a nomogram. So you can kind of think of it in the same way as a baby growth chart. I'm sorry, this is quite small. I'm not sure how easy you can see this, but the hotter areas in the middle of the graph are the 50th centile of the whole population, and then you're going out in centiles in terms of the phenotype that you're looking at, and in this case it's hippocampal volume that you're looking at. And then the red dots on these graphs are brain health clinic patients, which of course can be plotted straight on top of the Biobank background. And first of all, you can see that the patients tend to be in the lower half of the centiles of the population, exactly as you would expect. But probably the more obvious thing that you would have spotted as soon as the graph came up is that most of the brain health clinic population fall off the end of the age range of the Biobank. And so one of the things that we're working on now in a project, the BIRAX project, in collaboration with colleagues in Israel, and this is being led by Heidi Hansenberg, is collecting a sample of people who are in that much older age, the oldest old age range, and so that we can make more, so that we can extend out those nomograms and cover the whole age span. Again, hot off the press, we've only just had the diagnosis and ApoE information, but we can have a kind of sense check to make sure that the data that we're deriving with these pipelines looks sensible in relation to the things that we already know. So the graph on the left here is showing hippocampal volume according to diagnostic group. So you can see the Alzheimer's disease group having the lowest volumes, the non-AD dementia being very similar, MCI being sort of in the middle, and then the group with no diagnosis have the largest hippocampi, exactly as you would expect. And then the mood and anxiety disorder group looking pretty similar to the no dementia disorder, no diagnosis group rather. And then on the right hand side of the slide here, you're seeing the hippocampal volume again by genotype group, and again it's exactly as you would expect. So we have smaller hippocampal volumes in the E4 carriers and particularly in the E4 homozygotes, and the largest volume showing up in the E2, E3 group. And of course, you'll be thinking that there's an interaction between which groups the patients are in. There are more E4s in the Alzheimer's disease group, and so that's sort of reflected in this as well. So I've shown you sort of a taster really of the sorts of research that we're just starting to be able to do with using the Brain Health Clinic data. But the ambition for the Brain Health Clinic is really that it's infrastructure that enables a lot of different sorts of research. We're just starting now to be able to do targeted recruitment for studies and trials using the information that we're collecting, where consent's being given, so that we can identify the right people for the right studies at the right time. We are starting, as you've seen, to think about how we use this infrastructure for novel biomarker evaluation. The imaging things are kind of baked in, but we're also exploring ways of incorporating more cognitive and digital assessments. Ludovica Grifanti again is really leading on the radiological decision support tool development. And sort of separate from me and my team is also the beginning of research that's sort of outside my typical sphere, but relating to the care pathways and implementation and patient experience, which again is important for the whole package. We are not the only people to be thinking about brain health clinics. There's a number of clinics springing up around the country, and we're sort of forming ourselves into a network. At this point, there are a number of different sort of motivations that have led to brain health clinics, but fundamentally it's all about trying to diagnose well, diagnose early, and incorporate these sort of personalized prevention plans into the workflow, so that particularly those people with the earliest signs of cognitive decline were able to offer packages of support around those modifiable risk factors in the first instance, but again thinking about the trials for disease modifying therapies coming down the line, which don't feel like they're so very far away now. And this brain health clinic network is sort of a key part of the Dementias Platform UK's plans to develop a trial delivery framework to really accelerate the availability of infrastructure and patients for the trials that are needed. So the brain health clinics provide a kind of constant flow of patients who are well phenotyped coming through these clinical settings, and that's sort of added to the large number of individuals, primarily healthy individuals, who have been sort of well phenotyped as part of existing cohorts from around the UK, and were being consented for recontact for trials, and together these create the opportunity to again get the right individuals into the right trial at the right time. So by way of summary, really, this slide shows you the various ways in which I think the brain health clinic is a kind of a win-win-win. So within the clinical setting, we're getting excellent feedback with regards to the clinical report that we develop. We're reducing appointment times by a substantial amount, and changing mindsets in the clinical setting about what patients are able to tolerate and what's useful for patients. The patients themselves, we've had excellent feedback, and they are getting, of course, a very thorough assessment and the potential for earlier and more accurate diagnosis. And then for the research, we've got more than 90, 80 and 70% consent for data, additional assessments and recontact respectively. We think this is providing a really representative data set with in comparison to the data we're used to using, and it's enabling this sort of precision recruitment for trials. There's an additional win, which is particularly in our context, where the healthcare leadership of Oxford Health NHS Foundation Trusts are really embedding research culture into their services a key priority. So this is a model for how that can be done, which is likely to be taken up in various other ways. But of course, there's still plenty to do. This was a pilot. There was a lot of work to conduct a pilot study. Nevertheless, there's all sorts of ways you can easily imagine sort of augmenting what we've done so far by incorporating more cognitive phenotyping, more digital phenotyping. We're very anxious that the next thing we need to do is incorporating biofluids, so particularly blood sampling and CSF sampling where we can, and we've got new industry collaborations who are interested in funding that bit of the process, because it's hugely advantageous in terms of then being able to recruit the right people to the right trials. I've talked about the trial delivery work that we're doing with the Dementias Platform UK. And of course, we have in mind how we scale this, because at the moment, this is still available only to a relatively small population of people who happen to live in South Oxfordshire. So scaling both within the trust, but also out into through the network, and also out into partner sites around the UK, where our sort of diversity can be increased by those who don't live in rural South Oxfordshire. And that's all I have for you today. I just want to say a massive thanks, as doing this work has obviously included a huge number of people, a large number of stakeholders. A massive thanks to the whole team who have made this work happen so far, and thanks to you all for your time today.
Thank you. It was a wonderful presentation, Claire, and very clear. I see a couple of questions already, and the first question is actually from Brian Mackay, and he's asking for the 4% of early onset Alzheimer patients who visited the BHC, are there mutation carriers in there? So I think it's targeting towards APP and presenilin.
Yeah. So that's not something we've assessed so far. We have the DNA of those who consented for that. We have it sort of safely stored because until we've got enough of it, really, because as you see, we're only dealing with a relatively small number of individuals for now. So we've ApoE genotyped them because that was the obvious thing to do. But no, we haven't sort of gone for the more substantial genotyping yet. We were actually kind of a bit stuck. We nearly did. We nearly did the Biobank genotyping just because we want to increase the sort of overlap between what we're doing and the Biobank. But then there are some reasons to not go for that panel because it's not necessarily the ideal one for dementia. So actually, I kind of need you genetics experts to help with what the best thing to do with those data are and at what point to do it, because of course it's a small n at this point. Yeah,
definitely is important. But if you look at the interest, so if are there patients with interest particularly to their genetic background or rather to the environmental prevention, because that is one of the hookups, right? That they want this personalized preventions. Do they ask you about that or?
So we do get asked by patients. Of course, we're only collecting the data when they see us. And so it's only when they get to the doctor a little bit down the line that they have to handle those questions, not us as we're collecting it. But I think there is a sort of, there is a groundswell of work starting to happen in relation to what you do. When if you are able to identify risk factors, what is the best way of communicating? How do you sort of stratify people in terms of what information they most want, what information is most useful, what's going to be least harmful, etc. And again, that's not something that we've kind of broached yet, but we'll be looking to others who are doing that sort of research for the best ways of doing that. But again, because we are in effect an assessment centre, it's not until they get to the clinic that sort of conversation can happen.
Yeah, I understand, but you could make an inventory of that, right?
Absolutely, yeah.
All right, there's another question from Sana Suri. Really finds it exciting stuff, but wonders whether you had the possibility to look at the comparative CT versus MRI.
Yeah, good question,
and
Sana.
The added value of that for the hippocampal volumes.
Yeah, it's a really good question, and that's actually very much part of what Ludovico Grafanti is doing in her fellowship, because we kind of do need a direct comparison between the two, so that we can assess the extent to which having an MRI is really changing the diagnoses. Because we have removed the CT from the people that we're seeing, we don't have a direct comparison, but we can look across at very equivalent patients who are not the ones who haven't been able to come through the brain health clinic and look for sort of profiles of what the diagnoses end up being. But I think it's also important to recognize that alongside collecting these data and putting these data, making them available to clinicians in this clinical report, there's also a kind of piece of work to do in terms of how best to use that information. So there's a kind of whole education piece that's needed there, which again is work that Ludo is leading on going forward. So yeah, kind of early days, but we're thinking about how we do that.
Yeah. And are you thinking of sending people for PETs at all, or?
So that's not
old topic in the DRO, of course.
So I think that's not something we can necessarily offer by in routine at this point. But of course, the targeted recruitment that we'll be able to do means that we'll be able to identify, a lot of these patients actually might well end up having PET because they'll get recruited into the studies where PET is a part of it. But no, with our current setup, sort of routine PET isn't an option at the moment.
Yeah. No, but it could be in subgroups, right? Then there is a question of Klaus Edmeyer on the great work, but the question a clinical question, what is the added value? Yeah. And then both on the field of imaging and neuropsychology.
Yeah. Yeah, absolutely, Klaus. And again, it's sort of a similar response to my response to Sana, and the added value, the clinical added value is obviously kind of paramount in terms of how we convince commissioners to pay for this sort of work. It's something that we're trying to collect baseline data for at the moment. At the moment, we don't have a way of doing that head-to-head comparison, as we said, because we haven't run this as a kind of randomized trial, but nevertheless, we do need to find ways of comparing and contrasting between equivalent patients who are coming through the non-brain health clinic route at the moment, so that we can do that. We've got kind of anecdotal evidence of the confidence that the clinicians have of making diagnosis on the basis of the report that we had, and we've got one or two kind of nice N of one anecdotes of individuals who actually. And I have one in particular in mind, a patient who actually incidentally did end up having a CT scan and an MRI scan. So in that case, the clinician was able to say on the basis of the CT scan, they would have made one decision, but actually the MRI scan really, or wouldn't have been able to make a decision. And the MRI scan helped them to feel more confident about the diagnosis that they were making. But, you know, sort of formalizing that is obviously a critical next step for us.
Don't see any more questions, but my question was, it seems to be very important to have a cognitive screening also, right? And an in-depth one. So that is being added now with Kia's group and Nahid.
So we're doing the ACE3 as the cognitive measure that the people are getting as they come through the door. One of the key things that we are asking ourselves is about the triaging process. So at the moment, our triaging is done brilliantly by a trainee psychiatrist within the South team, who has become a real expert in identifying the patients that he thinks are most likely to benefit from coming through the Brain Health Clinic. And he makes that decision based on his clinical expertise, but it's kind of a combination of frailty and his assessment of their cognitive function based on what comes from the GP, and also he talks to them on the phone. So he's trying to identify the people he thinks that the Brain Health Clinic information will be most useful for. But, you know, as we're going forwards, the question of whether there should be some kind of screening that's happening there, so that we're able to identify, you know, really sort of direct this service towards the people who are at the earliest stages of cognitive impairment, where the information is most likely to be more helpful. You know, that's a kind of, those decisions are things we're thinking about at the moment. But yeah, there's obviously loads of more things that you could plug in, and it's just a matter of kind of prioritizing those as we go forward.
Yeah, but I can also see that in addition to that, the follow-up of the patient, yeah. In the long run, you would like to have a baseline cognition also, in order to, you know, the usefulness of the cohort may improve enormously.
Yeah, I think that's really important. And again, high priority for the next phase of the project is being able to collect outcomes on a kind of ongoing and longer term basis. So at the moment, we're extracting from the clinical record the diagnosis that happens, you know, within a couple of months of patients coming through the clinic. And we have plans to sort of check back in on yearly intervals to see when diagnoses change, but that will be very imperfect. And so we ideally want to be plugging people into ways of following up, probably remotely, probably via the relative, as best we can, and trying to collect that information in that way. I actually think that's possibly even more important than the bloods in terms of the value of this information that we're collecting at the moment. I've just seen that Anya asked about longitudinal imaging. We're not planning on longitudinal imaging, you know, as part of the sort of routine brain health clinic, Anya, unless people get re-referred back in to the service, of course. But as you see, we've got a good number of these patients who've consented to future studies and trials. And so if somebody had a particular study that they wanted to do, which would be following these patients up in over a particular time period, then we've got everything set up to make that easy to do.
Yeah, there's another question from John Stein. Does the Brain Health Clinic build upon the Optima study at all?
Yeah, I mean, I guess John, that it's not a million miles away from the Optima study. The Optima study was a study rather than sort of embedded into the clinical service in quite the same way. But of course, it was also collecting high quality, and of course, there were several phases of the Optima study along the way. So yeah, you could think of it as being Optima 2022. What we think is that we're collecting a much more kind of representative. It's this kind of alignment with the clinical service that makes it different. So we're offering this to everybody that's coming through. So it's the data that we end up being able to use in the research setting doesn't come from a research cohort. It comes from a clinical population. And again, thinking of those as individuals that we can compare with the kind of mass of the UK Biobank means that it sort of we think gives us kind of real world validity for some of the kind of modeling that we want to do with that developing predictive models, etc., from the UK Biobank. So yeah, I don't mind it being thought of as Optima 2022 with some bells and whistles.
All right, Klaus came back with the point that would it be nice to persuade the GP commissioning groups and the successors to that such follow-up is better than the GP follow-up?
Yeah, well, I think it probably would be, Klaus, but I'm going to let you fight that battle.
Yes, to bring it up in a gentle way, I guess. Right?
Yeah. If you have any ideas about how we could do that, I'd be delighted to discuss with you.
Yeah, well, because part of it, the GPs are not that keen following up, I guess.
Actually, I think
their degrees of freedom are so limited to follow up.
Yeah, in a less facetious way, I think the link with general practice is actually really key, and particularly as we're all thinking about this earlier stage. So you're not really in secondary care then, you're in the primary care world, and how we interface this sort of, particularly that risk management, the personalized risk reduction things, that I think that sort of belongs in primary care, but how we embed that or engage with it, I think that's another way in which we need to think going forwards.
I think Klaus makes the same point. He agrees with me. They may be grateful instead of being offended. And maybe that is grateful, but I think the follow-up on the blood-based biomarkers for the ATN criteria, amyloid tau and neurodegeneration, that may be, I think, very interesting, and definitely you would like to connect that. I know Masud wants that with the cognitive function decline. Yeah, definitely. If there are no more questions, I had the last question. So you don't have a control group in the setting, and secretly the Amsterdam group always did. They have these cases that they said, well, they didn't have any dementia, and therefore they are the controls. But of course they came in.
Well, I mean, we have a few of those. The ones who don't get a diagnosis are that category. But we are actually collecting a control group. So I skimmed over it fairly rapidly. But the BIRAC study is actually all about collecting age-matched controls from the general population. It's interesting because we really the Biobank takes us up to sort of age 80-ish, and then but we really need some older people than that, and so we're just starting a campaign called You're Never Too Old to try to engage with people who will think that their brains are less useful for research, but in fact those are the ones we really want for this. And so we will in fact be collecting a sort of a small but direct control group for you may also think just take the spouses maybe, because they are in anyway. Yeah, absolutely. They come together, which is also very nice to have them on the same. Yeah, and so where relatives have come along, we have a sort of registry of people who are interested, and the relatives who consented for recontact are all in there. So that we do those as well. We are doing those as well. Well, thank you very much, Claire. We're up to the hour, and it was a fantastic presentation. Thank you. Thank you very much. Thank you.